Go ahead, Vlad.
So I've known Betsy since 1965. That's the year she came to Columbia to work with me as a postdoc. Should not surprise anyone who knows Betsy that she was a fantastic postdoc. Therefore, when I was being recruited to Mount Sinai, I insisted that Betsy be recruited together with me. She got an appointment at Mount Sinai and performed spectacularly well, so that by now she's a full tenured professor at the Mount Sinai School of Medicine. Betsy had a magic touch that whatever project she touched, she turned it into gold. She turned out to be an excellent biochemist without having a background in biochemistry. She purified and established the structure of true neuropeptides, myomodulin and baccae. She localizes neuropeptides to motor neurons and demonstrated their bioactivity. At the theoretical level, Betsy introduced therefore the notion of intrinsic modulation, which became popular and is widely used by now. Over the years, it's important to point out that it's been believed at the time when she was started working on peptides that they were released only under conditions of extremely high firing. Betsy then recorded motor neuronal activity during feeding behavior and demonstrated that this is not the case, and showed that peptides are released with normal range of neuronal activity. I could go on and on about specific things. Many of them are really important, like the ones I already mentioned, but I think it's best to give voice to Betsy to describe the things she wants to us to know about her work. Betsy, it's your turn.
Thank you, Claude. Thank you for that really kind introduction. And thank you guys for asking me to speak in this series. It's really an honor. Okay, so today I'm going to talk about intrinsic modulation in the feeding system of Aplysia. So there'll be two parts to this talk. So I'm going to start by describing experiments I did as a postdoc. So these experiments studied peptide co-transmission in a neuromuscular system. So this work was mostly done in the Center for Neurobiology and Behavior at Columbia, and roughly between the years 1985 and 1995. And then secondly, I'll talk about experiments that are ongoing that study intrinsic modulation in the feeding circuit. And this work is mostly being done at the Mount Sinai Medical Center. So the first work that I'm going to talk about, the neuromuscular work, was primarily done in Claude Weiss's lab. So I was a postdoc with Claude. Other people, Scott Hooper, Mark Miller, Fred Willem, and Claude himself did some of the work. Some of these experiments were also done in collaboration with Irving Kupferman's group. Renata Tenenbaum did all the early reverse phase HPLC, and the peptide sequencing was done by Marianne Koltz at the Howard Hughes Protein Chemistry Core at Columbia. So first, a little background. So at the time my project started, it was well established that peptides coexist with small molecule transmitters, and it was clear that this was the case in both vertebrates and invertebrates. Also, the Jans had demonstrated co-transmission in the sympathetic nervous system of the bullfrog. There were still a lot of questions, though, that remained unanswered, such as under what circumstances does peptide release occur? So when my project started, peptide co-transmitters in invertebrates were primarily being studied in insects and crayfish. So mollusks were a bit behind. So in mollusks, peptide hormones had been characterized like ELH. Also, several cardioactive peptides had been identified that were good transmitter candidates. And in particular, there was a lot of interest in the SCPs, the small cardioactive peptides. And there are two SCPs, SCPA and SCPB, that are present on a single precursor. So the system that I used was the system that you see here, the ARC neuromuscular system. So the ARC, the accessory radula closer, is a muscle that Aplysia used during feeding. So this muscle then is innervated by two cholinergic motor neurons, B15 and B16. And so this system had been characterized before my project started by Josh Cohen working with Claude and Irving. So an early question then that my project addressed was, do the ARC motor neurons contain the SCPs? So to address this question, we used a technique that was developed by Philip Lloyd working with Claude. So Philip was Claude's postdoc before I joined the lab. So in effect, I replaced Philip. So with this method, we identified motor neurons physiologically and injected them with fast green dye. We then incubated ganglia in seawater that contained radio-labeled methionine. So methionine is an amino acid that's present in the SCPs. So the idea here then was that if neurons synthesize SCP, they'll take up this radio-labeled methionine, synthesize radio-labeled SCP. So Claude then dissected neurons out of the ganglia and put them in a solution that contains synthetic SCPA and B. So this whole mixture then was subjected to reverse phase HPLC, and obviously we were looking for coelution of synthetic material and radioactivity. So what we found then was that the SCPs were present in one of the motor neurons, B15. The other motor neuron, B16, was negative. So kind of an interesting thing with this, the technique that we were using was that potentially negative results could be interesting. So some negative, in one case, you could get a negative result if a neuron didn't synthesize anything radio-labeled that had a peptide-like elution time. And that, of course, wouldn't be such an interesting result. But another possibility is that you could find that a neuron synthesized something radio-labeled, but something that wasn't one of the SCPs, something that had a peptide-like elution time, but wasn't one of the SCPs. So this result then is obviously potentially interesting because it suggests that a neuron could contain a novel peptide. So this is what we found then for B16. So B16 was negative for the SCPs, but it did synthesize radio-labeled peptides. Also, when we looked at B15, B15 synthesized radio-labeled material that did coelute with the SCPs, but also material that didn't. So this suggested that there were other peptides in B15 as well. So an obvious question then became, what are these other peptides? So to address this question, we decided that we wanted to purify everything that we could that was bioactive in the ARC system. So to do that, we decided to purify peptides from terminals, from the B15 and B16 terminals. So terminals are obviously a good source of peptides. Also, the terminals of both motor neurons were attached to this one muscle, the ARC muscle. So with one fairly easy dissection, you would get material that's in both cells. So we dissected out the ARC muscles. We purified material using reverse phase HPLC. So we took out all these muscles, there were preparative steps, and we loaded the material onto the HPLC columns. So for the most part then, fractions of interest were identified by their bioactivity back on the ARC neuromuscular junction itself. So this is an example of what one of the bioassays looked like. So in this experiment, we were using B15. We did other experiments with B16, either motor neuron worked. So this is actually a slow record. So these are bursts of action potentials in B15. So this fraction then produced an increase in contraction amplitude. So this, of course, would have been a fraction of interest. So fractions that were interesting, that were bioactive, that were of interest, were rechromatographed, and so on until we got material that was pure enough for sequencing. Okay, so to get material that was clean enough for sequencing, it took a number of passes. So the number varied depending on the peptide. The range was 5 to 7 passes. So as a result, we had to assay material literally from thousands of HPLC fractions. Testing each fraction was time consuming. We had to take an aliquot from each fraction. We were, there's sequential chromatography, so we didn't want to bioassay the whole thing. So we took an aliquot from each fraction. We had to dry them down. We couldn't put solvents on the neuromuscular junction. Fractions had to be reconstituted and then tested in this assay that relied on intracellular stimulation and reporting. So it wasn't a trivial assay. So as a result, this whole thing was a lot of work. In our opinion, though, it was totally worth it. It was very rewarding. We did, in fact, get sequences. We pulled peptides out of the ARC system. So some of the peptides we sequenced were, not too surprising. So we pulled out SCPA and SCPB, and we already knew that the SCPs were present in B15. We also found that formaframide was present in the ARC system, and formaframide was a peptide that had previously been characterized. So some of the peptides we pulled out then had previously been described. Other peptides that we pulled out, however, were novel and had not been described. So 9 of them subsequently turned out to be members of the myomodulin family, 3 of them members of the buckalin family, and 3 others members of the FRFMI family. Okay, so to purify things, to get things pure, it took a number of steps. There were losses with each step. So actually, when we got to the end and to the sequencing, there wasn't very much material left. So actually, the sequencing was very difficult to do, which was why I made a point of saying it was done by Marianne Colts. She really did a fantastic job. It really wasn't very easy. So we certainly could have made mistakes with our sequencing. Fortunately, we didn't, however. So subsequently then, precursor proteins for our novel peptides were cloned. And this work was done to a large extent by Mark Miller working with Jürgen Brosius. So this is one of Mark's papers. So this is the characterization of the buccal precursor. So the cloning then was important because it confirmed our sequences, and it also identified less abundant peptides that we couldn't purify. So here I told you that we pulled out 3 buckalins. As it turned out, there weren't just 3 buckalins. So this is a list of the buckalins that are on the buccal precursor. There's something like 18 or 19 of them here. Okay, so we had novel peptides. Then the next question was, are they in B15 or B16? So to answer that question, we use the technique that I described for you. So in some cases, we were again able to do experiments with methionine. So methionine worked for the myomodulins. They have methionine. Other novel peptides didn't, so we used other radiolabeled amino acids as well. So what did we find in the end? So one of the novel families, the buckalins, were present in both of the ARC motor neurons, both B15 and B16. As I said, the SCPs were in B15, not B16, but B16 had the myomodulins. Now, if you're wondering what happened to formaframide and the FRFMI peptides, it turned out they're in the terminals of sensory neurons that also integrate the muscle. So they weren't in either one of the motor neurons. Okay, so a lot of this work then focused on the ARC motor neurons, but these weren't the only cells that we looked at. So we also looked at other feeding motor neurons as well. And so what we found then was that other feeding motor neurons contain peptide co-transmitters. This wasn't something that was unique or unusual just for B15 and B16. So most of the feeding motor neurons we looked at also had peptide co-transmitters. We also found that peptide levels in motor neurons were lower than levels in neural secretory cells, such as the bag cells. So as a result of these factors, probably other factors as well, certainly at least initially at Columbia, our work got sort of a lukewarm reception. So that the idea was kind of, okay, you're showing that there's peptides in a lot of these cells, potentially in low levels. That can't be very interesting. So we, of course, thought that it was. And we wanted to establish that peptides were playing an important role in normal behavior. So to do that, we thought it's going to be important to show that the peptides are released when the motor neurons fire at physiological frequencies. So to do that, of course, we had to characterize the firing frequencies of the ARC motor neurons. So for these experiments, then we used a technique that was very simple in principle. So we took a very fine wire and stuck one end of it in the ARC muscle. So in this picture, this little band here is the ARC muscle. So we stuck one end of the wire in the ARC muscle, glued it in place, sewed up the animals, and let them just engage in normal feeding behavior. So we found then that the firing frequencies of the ARC neurons did depend on the nature of the behavior and were different for the 2 motor neurons. There was one ingestive response that we looked at a lot, which was a swallow. So we found that the B15 firing frequency during swallowing was between about 7.5 and 12 hertz, the B16 firing frequency between 15 and 20 hertz. We also found that the motor neurons fired in bursts of about 3.5 second duration with an interburst interval of about 3.5 seconds. So when we looked at these data then, we thought, okay, there's sort of bad news and good news here. The bad news was that the firing frequencies looked kind of low, particularly B15. And this was potentially a concern because it had been suggested that peptides are only released at high firing frequencies, and high firing frequencies in particular meaning 20 hertz or above. And this was an idea that appealed to people because it was known that peptides are present in dense core vesicles, and dense core vesicles are generally not in the active zone of the synapse. So the idea is, right, to get peptide release, you have to have an increase in the intracellular calcium concentration that is going to extend beyond the active zone. So how would you get this kind of an increase in intracellular calcium? Well, one way you can certainly do it is with a high firing frequency. So sort of the bad news then was potentially these firing frequencies look low, particularly B15 firing at 7.5 to 12 hertz. We also thought, though, potentially there's sort of good news looking at these data, though, and that the interburst interval was so short, because it was known at the time that PTP, presumably mediated by potentiation of acetylcholine release and residual calcium, could be induced with this type of duty cycle. So we thought, okay, so here's another mechanism where you could have an increase in intracellular calcium beyond the active zone. So maybe we don't have the high firing frequencies, but we have the short interburst intervals. Maybe that's what's inducing the peptide release. Okay, so to see if that was true, we of course wanted to look at peptide release. Initially, we had to do it with using indirect techniques because we didn't have the tools to do it directly. So when I say indirect, more than one method was used. Probably the most successful method was measuring cyclic AMP in the ARC. So doing experiments that took advantage of the fact that SCP exerts cyclic AMP mediated effects, and acetylcholine doesn't do that. So we stimulated B15 and looked to see whether there was an increase in cyclic AMP in the ARC. So we were doing these experiments, or we did some of these experiments, simulated physiological frequencies, and found, yes, we were getting, for example, cyclic AMP in the ARC. So that we were very happy with that. This work, however, while we were doing our studies, this work was also going on, or this type of work was also going on in Philip Lloyd's lab. So to come back to Philip, Philip had left Claude's lab and finished his postdoc, started his own lab, and was at the time also looking at peptide release in the ARC system. And in Philip's lab, this work was being done by Matthew Wim. So a question that was of interest to Matthew and Philip was, is release in the ARC system frequency dependent, as had been demonstrated in other systems? So Matthew and Philip found that it is, as you can see, for example, over here. So this is a figure from this paper. And this is an experiment where the cyclic AMP in the ARC is being measured. So you can see the frequency dependence if you compare this bar to this bar. This is 20 hertz, 17. You can also see the frequency dependence if you compare this bar to this bar. This is 15 hertz and this is 12.5 hertz. If you're wondering why this 15 hertz bar is bigger than the 17.5, it's because the interburst interval is different. So this is a shorter interburst interval than this. Okay, so this is showing that release is frequency dependent. So the problem here, however, is that only one of these sets of parameters is physiologically relevant, and that's this one, this at 12.5 hertz. And so what Matthew found then was very little to no cyclic AMP in the ARC with this frequency. So actually, so these sets of results were presented at the same Society for Neuroscience meeting. So what happened was I got up and said, with these parameters, you get release, or you get cyclic, anyway, you get cyclic AMP in the ARC. And Matthew got up right after or right before and said, with these parameters, you don't. So there was, the results were directly conflicting, which was rather awkward. And until Claude got together with Philip and discussed, we discussed, exactly how the experiments are being done. So what it turned out was that the difference was temperature. So Matthew's experiments were done at room temperature and my experiments were done at a colder temperature. So later, as I'm going to tell you, we were able to measure peptide release directly and confirm that temperature, peptides are not released at, or very little peptide is released at warm temperatures in the system. Okay, so as I mentioned then later, we were able to measure peptide release directly. We now actually means Fred. So Fred developed a method for doing this in collaboration with David Price. So in Fred's experiments then, motor neurons were intracellularly stimulated, of course, using physiologically relevant parameters. peptides were released from the terminals of the motor neurons, which are attached to the muscle. And this preparation was set up so that the muscle was outside the recording chamber. So you might think that the muscle is going to dry up. Very fortunately, though, this muscle has a little artery, and so Fred was able to perfuse the muscle through the artery. Also, something that's not shown here is that this whole thing, there was Vaseline on this, and it was wrapped in parafilm to keep the muscle from drying up. So peptides were released into the perfusate, drops formed, and then these drops fell right into test tubes, which were used to run RIAs. So we measured the peptides with RIAs. So the important thing here was that the drops went directly into the tubes, so there's no transferring of material and no losses due to transferring. Okay, so with this method, Fred was able to directly measure peptide release and show that peptides were released when physiologically relevant firing patterns were simulated. So confirming what we showed with the indirect methods. So here's an example of one of Fred's results. So this is from a single animal. In this particular case, Fred was measuring SCP release from B15. Fred also did experiments where he measured buccal release from B15 and experiments where he looked at buccal and myomodulin release from B16. So what's plotted here is femtomoles of peptide per drop. So that's what each one of these dots is, femtomoles of peptide per drop. So as the experiment begins, there's no peptide release because there's no stimulation. So the neural stimulation begins here where you see the bar. So the purpose of this experiment was to see what the impact of changing the interburst interval was. So Fred started out with a relatively short interburst interval of 3.5 seconds, which is physiologically relevant, and that did in fact induce peptide release. Fred then lengthened the interburst interval, release goes down, and then it comes back up. So on the bottom, you see group data. So there was a lot of variability, so data had to be normalized so that it could be pooled. Also, what's shown here is release per action potential because there are different numbers of action potentials with these different paradigms. And there's 2 lines here because what's plotted is both buccal and SCP release. And so you can see as the interval gets longer, there's less release. Okay, so studies of peptide co-transmission in motor neurons in the feeding circuit then demonstrated that peptide co-transmitters are present in many motor neurons, established that peptide co-transmitters can be released during normal behavior, and demonstrated that release doesn't require a high firing frequency. It can occur when the interburst interval is short. So a next obvious question, of course, is going to be what do peptides do? So
Betsy, can I interrupt you for 1 second?
Yes.
So are you saying that the burstiness of the stimulus is still required? I mean, is a requirement, and that the interburst interval determines how much is released? Or could tonic firing also release in this case?
No, tonic firing could. No, it's not. I'm not saying that the it has to be bursty. That is just the mechanism that's used in the system. So like high firing frequency will do it. And you know, if you stimulate it at 50 Hertz for tonically, you get a lot of peptide release, sure.
Betsy,
Okay.
since you were interrupted, can I continue
Sure.
to interrupt?
Of course.
I know the answer to this somewhere inside my brain, but a few minutes earlier when you mentioned that at room temp or for aplasia high temp, there's no peptide release. Is that particular for peptides, small molecule transmitter release? Does that still occur at those higher temperatures?
So, I mean, so we never looked at this really, what's going on. You know, why are we not seeing the release? I mean, Claude and I were just talking about this the other day because it's kind of interesting given some of the results that you guys have about the importance of peptides at high temperatures. We don't really know. There is acetylcholine released. I don't know that it's the same amount, but there is acetylcholine release. I mean, you can get a muscle contraction at the high temperature. Our best guess is that what happens is that the action potential duration is changed. And that's why there's, but we didn't look at it, honestly. We weren't so interested in it. And I mean, the effective temperature wasn't, we just didn't look at it. We should have.
Well, there's always now.
Yeah, yeah. Unfortunately, we're not set up to do that. We don't do radioactive, and nobody does radioactivity anymore. We don't do RIAs.
All right, thank you.
Yeah. Okay, so what I think I'm going to do, so instead of talking about what the peptides do in the ARC system, I'm going to move ahead and talk about what they do in the feeding circuit, because I think that that's probably going to be of more interest for this audience. But before I do that, I want to just acknowledge that I'm skipping a whole body of work. So for example, work that was done by Bill Probst, Scott Hooper, or Harish and Jorg Heyerhorst that looked at second messengers activated and proteins phosphorylated in the ARC system. Also work that was done by Colin Evans to characterize the radula opener system, and work done by Vlad Brzezina, Ashad Kozak, and Marcia Scott that characterized currents induced and modulated in the ARC and opening muscles. Okay, so what I am going to do now, though, is move ahead and start talking about modulation in the feeding circuit. So this work has and is being done in the Ecclesia Research Group, which is primarily at Mount Sinai. So currently, the laboratories in this group are my laboratory, John Jing's, and Claude Weiss's. And I say we're primarily at Mount Sinai because John Jing's primary appointment now is at Nanjing University. We're very fortunate, though, that we've been able to continue to work together. Okay, so before I talk about the work in the feeding circuit, I have to just tell you a little bit about feeding in Ecclesia. Tell you that to feed, to grab food, Ecclesia use a structure which is known as the radula. So you see the radula in this photograph. So this is the head of an Ecclesia. This is the mouth, which is open. And this yellow thing then is the radula. So here the radula is closed. So here you see how Ecclesia use the radula during an ingestive response. So this is the side of the head. This Pac-Man-like thing is the radula. So during an ingestive response, the radula protracts open. It then closes on food and retracts, pulling the food into the buccal cavity. So in addition to ingestive behaviors, there are also egestive behaviors. And so this slide shows a major difference between the two. So in both cases, you have protraction, retraction, protraction, retraction. A major difference is in the timing, relative timing of radula closing. So during ingestive behaviors, the radula closes as it retracts, which pulls food in. In contrast, during egestive behaviors, the radula closes as it protracts, and this pushes food out. There are also behaviors where closing isn't restricted to either protraction or retraction. These are referred to as intermediate behaviors. Okay, so feeding in Ecclesia is a CPG-mediated behavior. The CPG is reasonably well characterized, so we can trigger feeding motor programs in vitro. So there's more than one way that this can be done. One way it can be done is by stimulating neurons that are in the CPG. So when programs are triggered this way, protraction interneurons are generally used for this purpose. So there's reciprocal inhibition between the protraction and retraction interneurons. Additionally, protraction interneurons provide slow excitatory input to the retraction interneurons. And this slow excitation is important because there's very little post-inhibitory rebound in the system. So if you inject current into a protraction interneuron, you generally get both the protraction and retraction phases of the motor program. There's very little slow excitation from the retraction circuitry back onto the protraction circuitry. So to keep activity, to make activity repetitive, you have to keep driving the protraction circuitry. And this mirrors what you see in the intact animal, in that feeding in the intact animal is episodic. Okay, so one way you can trigger activity is from within the CPG. If you do that, if you take a rested preparation and trigger a cycle of activity this way, generally the cycle of activity will have intermediate characteristics. And you can see that, for example, in this cycle. So this is from a paper of Michael Simscalchi's. So this cycle of activity was triggered by stimulating the protraction interneuron B63. The bottom two traces are extracellular recordings that were used to recognize protraction and retraction. What's important then is looking at this intracellular recording from a radula closer motor neuron. And what you see is low frequency activity during both protraction and retraction. So that's what makes this cycle of activity intermediate. So if this was egestive, you'd have high frequency closing and protraction. If this was ingestive, you'd have high frequency closer activity and retraction. So closing during retraction, pulling food in. Okay, so this is intermediate. Okay, so how do you get defined activity or activity that's defined as ingestive or egestive. So this can be done by stimulating CPG inputs. So a well-characterized ingestive input is a command-like neuron, CBI2. CBI2 stands for cerebrobuccal interneuron. So CBI2 then gets excitatory input from sensory neurons that are activated when food contacts structures such as the lips and the rhinophores. A well-characterized egestive input is a branch of the esophageal nerve. We usually just label it EN. So the EN then contains the processes of sensory neurons that innervate the gut. And one way you can trigger egestive behavior in aplysia is by forcing an inedible object into the gut. Okay, so what do the CPG inputs do to the inputs due to the CPG? One thing they do is excite the protraction circuitry. So produce fast EPSPs in protraction interneurons. And both CBI2 and EN do this. So this is important, of course, to initiate activity. Additionally, CPG inputs contain modulatory peptides. So CBI2, which is, by the way, a cholinergic cell, contains FCAP and CP2, the peptides FCAP and CP2. When you stimulate the EN, you see release of a number of peptides, so the SCPs, FRFamide peptides, and an aplysia version of neuropeptide Y. So it's two different sets of peptides here. Okay, so what do the peptides do then? A number of studies have looked at this and have strongly suggested that they play an important role in configuring motor activity. So these studies have taken a variety of approaches, triggered motor activity and put on exogenous peptides, looking at effects of endogenously released substances, and looking at where in the circuit peptides are acting, and to some extent second messengers. Okay, now at this point I'm going to take you back and show you this figure, which I showed you before when I was talking about the ARC, the neuromuscular system. And so this figure, which shows that in the neuromuscular junction anyway, peptides are preferentially released when interburst intervals are short. Okay, so a question you could ask is, does the same thing happen in the central nervous system? So I'm telling you that peptides are important for configuring activity. So an obvious question to ask would be, does program configuration preferentially occur when the interburst interval is short? So the short answer is yes. So this was first demonstrated by Alex Proack working with Claude. So Alex and Claude did experiments looking at the effect of activating the ingestive input to the CPG, which is CBI2, also the egestive input, the EN. I'm just going to tell you what happens with CBI2 because what happens with the EN is similar in principle. So in these experiments then, what Alex did was he took arrested preparation and triggered a single cycle of activity with CBI2. So what Alex found was that this cycle had intermediate characteristics. So what's plotted here is the radula closer activity and protraction versus retraction. So you see low frequency activity, about 2 Hertz activity during both protraction and retraction. And this figure incidentally comes from a review, which is why there are no error bars. This is not the original figure. Okay, but this intermediate characteristics, and you can see that here. So this is an intracellular recording from a radula closer motor neuron, low frequency activity and protraction, retraction. Okay, so that was arrested preparation, 1 cycle with CBI2. Okay, so how do you ever get ingestive cycles? Alex found with repeated stimulation of CBI2 if the interburst interval is short, and that's what you see here. So this cycle 1 is this cycle again. This is the second cycle with a short interburst interval, third, and so on. And so what's happening then with these repeated cycles is that there are progressive increases in the radula closer firing frequency during retraction. So these cycles are getting more and more ingestive. So closing during retraction pulls food in. This activity is getting more and more ingestive. So what Alex found then is with repetition, program definition does occur, but this only does happen with a short interburst interval. So this phenomenon then has been referred to as ingestive repetition priming. And so as I mentioned, so this is ingestive repetition priming. This is what you see with repeated stimulation of CBI2. You see the same type of thing with the EN. So rested prep, single cycle, it's intermediate. If you repeatedly stimulate the EN with a short burst interval, you get egestive repetition priming. So you have both ingestive and egestive repetition priming. Okay,
Betsy,
sure.
Could you go back to that other slide for a sec?
Say that again.
Could you go back to the previous slide?
This one?
Yeah, this one. Yeah, I was just curious. I can't remember whether this was in the paper or not, but do you guys know whether there's a process in vivo for either of these systems, CBIs or the ENs, to be repetitively activated? Is there a sensory system that does this, do you know, or do they become intrinsically active, or blah, blah, blah?
Yes, yeah, sure. So yeah, so CBI2 receives, gets excitatory input from sensory neurons that are activated with food contact. So you can do experiments in an intact head kind of a preparation. Steve Rosen did this. And and also John Jing did some of this work as well. You can if you keep applying food, then you will get repeated activation.
Ah,
so
cool.
you can take a piece of seaweed and touch and then take it away, and you might just get a single or something. But if you keep applying it, it's the same thing in the intact animal too. You could get a single bite, or you can get a series of them.
Thanks.
Okay. So
Can I just ask there
Sure.
another question? When the animal is not hungry anymore and you present the food, is the sensory input still able to produce the feeding behavior?
I don't, ask me the question again. I don't think we know the answer, but so the animal is, say that again?
When you feed the animal well and the animal is not hungry anymore,
So satiated.
is the sensory input gated in some way?
Oh, okay. I'm just trying to think if we have any idea of that. I don't think there's any data that has ever suggested it's gated. So actually what happens, I'm just trying to think what's going to happen in that situation. So then you start getting a lot of signals from the gut, right? Because then there's distension and everything from the gut. So you get a lot of, you're going to get a lot of sensory input from the gut coming in. So you're going to get a lot of input to the CPG from the EN. So what's going to happen in this situation is the modulators that tend to make activity egestive are going to start acting on the CPG. So it's not the same thing as gating out. You know what I mean? Am I answering your question?
Okay. Yes.
But you're going to start having the impact of the other, from the other end of things. I don't think that we know for sure that there isn't any gating out. But.
Thank you.
Okay. So what do the peptides do? So in the CNS, they can be important for configuring the activity of the CPG. So a question then that's been of interest to us are what are the consequences of configuring activity this way? That is through transmitters that exert second messenger mediated effects. So second messenger mediated effects, you expect them to persist. So an obvious question you could ask is, do effects of repetition priming persist? So yes, they do. So the persistence varies. It determines by factors such as how the priming was induced, what circuit modifications you're looking at, and whether the priming was ingestive or egestive. So to some extent, when you're comparing ingestive and egestive priming, you're comparing apples to oranges. But in general, ingestive priming, the effects of ingestive priming tend to last longer. So the purpose of this slide is just to show you an example of persistence. So this is an experiment that was done by Matthew Perkins. And Matthew was looking at the effect of ingestive priming on the excitability of this particular neuron. So here's the control, here's the ingestive priming, excitability goes up, and it takes a while for excitability to come back down to control levels. So something like 40 minutes. So this is on the long side. The range here would be like 10 to 40 minutes. Okay, so the feeding circuit multitasks. So it generates both ingestive and egestive motor programs. So a question that's been of interest then is, how is persistence going to impact task switching? So maybe an obvious question to ask is, will it actually impede task switching? So in particular, this was first addressed by asking what happens when there's an attempt to go from egestive activity to ingestive. So putting the network in an egestive state and then trying to stimulate the ingestive input, what's going to happen? So this question was addressed by Alex, first addressed by Alex working with Claude, actually in that first paper that I mentioned. And what Alex and Claude found was that it doesn't happen easily. It does impede. So what Alex did in his experiments was started by repeatedly stimulating CBI2 just to characterize ingestive activity, just to show that these preparations were capable of generating ingestive activity. So those data are plotted here. This is radula closer activity in protraction versus retraction. So it's higher in retraction, closing and retraction. This is ingestive, okay? So this is just to show here, this is ingestive activity. Okay, the real question of interest here though is what happens when there's a switch, what happens when there's been a preceding bout of egestive activity and then you try to stimulate CBI2. So Alex did that, repeatedly stimulated the EN. Those data are plotted here. So that just shows that Alex did successfully put the network in an egestive state, right? So this closer and protraction is higher. So this is egestive. Okay, so really what was of interest is what happens if you stimulate CBI2 right after that, after this egestive activity. And so what Alex found was that this cycle of activity is egestive. So this has been referred to as negative biasing because usually if you take a rested preparation, stimulate CBI2, that cycle will be have intermediate characteristics. What happens after egestive activity though, is that this cycle is fully egestive. So on the next slide, you see another way of looking at negative biasing. This is an experiment that was done more recently by Flora Wang. So what Flora was doing was looking at repeated stimulation of CBI2 in the situation where there is no preceding egestive activity versus when there is. And these data then are plotted over here. So in Flora's graph, this is cycle number versus radula closer firing during retraction. So it's a measure of how ingestive things are. And so what you see when there's been preceding egestive activity, there is an impact. This is different than this. And incidentally, the dashed line here is approximately at the 4 Hertz firing frequency. This is approximately where contractions, where you get a functional closing. So what you're seeing sort of approximately here is that normally it would take 3 cycles to get to the functional closing, and with the preceding egestive activity, it takes longer. You're up to 7 here. Okay, so persistence can impact task switching in a negative way. That is, decrease the speed with which task switching can occur. A further question we've asked though is, does it always work this way? So what about a task switch in the other direction? So what happens if you attempt to go from ingestive to egestive? So the negative was when you went egestive to ingestive. So what if you flip things around and try to go ingestive to egestive? Are you going to see the same thing? Are you again going to see negative biasing? So this is a question that was asked by Andrew Dax working with Claude. And what Andrew found was, no, you don't see the same thing. You see the opposite. Actually, in this case, you see positive biasing. So what Andrew did in his experiments, cycles triggered with EN, the interburst interval is relatively long here. So there's relatively little priming. So these are intermediate cycles. Andrew then repeatedly stimulated CBI2 to make activity ingestive and then went back to the EN. So the point here obviously then is, how is this ingestive activity going to impact the cycle? So is this cycle after the ingestive activity going to be different than before the ingestive? And what Andrew found was that there is a difference. This cycle is different than this one. And this cycle is more egestive than this one, despite the fact that this is ingestive activity intervening. This is more egestive than this. And you can see that looking at the single cycle, the radula closer activity during protraction is higher here than here. And also group data here. So this is radula closer firing frequency during protraction, a measure of how egestive. So before, control, and then you have the ingestive activity, and it's more egestive afterwards. Okay, so in one direction, persistence impacts task switching in a negative way. So you see negative biasing. In the other direction, however, persistence has a positive effect. So you see positive biasing. So negative biasing is observed after repeated stimulation of the EN. Positive biasing is observed after repeated stimulation of CBI2. Okay, so to finish my talk, I want to just talk a little bit more about what happens with the repeated stimulation of CBI2 and what state you're in this situation. Okay, so with the repeated stimulation of CBI2, you see positive biasing. So as a result of this, you end up with a situation where there's more than one way that you can make activity egestive, right? So if you take a rested preparation, single cycle with EN, that's intermediate. So this cycle would be intermediate, okay? But you can make activity egestive by repeatedly stimulating the EN. If the interburst intervals are short here, this will be egestive, okay? So that's one way you could do it. Another way you could do it is by repeatedly stimulating CBI2 and then switching over to EN, this positive biasing protocol. Okay, so two different ways that you can make activity egestive. So a question then that was of interest to Flora Wang was, are the circuit modifications the same in both cases? And Flora found that they're not. So Flora's experiments took advantage of the fact that Andrew and Claude actually had already done a lot of work looking to see how positive biasing is mediated, and found that to a large extent, it's mediated by an increase in the excitability of an egestive interneuron, the cell B65. So what Flora was able to do then was to ask what happens with egestive priming. Is it the same cell B65 or a different cell? And so what Flora found was that it's primarily a different cell. It's this egestive interneuron B20. So there's a form of network degeneracy then in this situation. It's B65 in one case, it's B20 in the other case. Okay, so a question then that was of interest to Flora was, why would this be important that you use two different cells in these two different, you use a different cell in these two different situations? So what Flora's work showed, taken together with what Alex and Claude had showed earlier, is that it impacts how quickly there can be a transition to ingestive activity. So when egestive activity is mediated by an increase in B20 excitability, you can't immediately transition to ingestive activity. And these are the data that I showed you before. So repeated stimulation of the EN, this is the B20 situation, you go to CBI2, and you get an egestive cycle of activity. So you cannot immediately transition to ingestive activity. So a question then that was of interest to Flora is, will you see the same thing after positive biasing? And Flora found that you don't. So Flora's question was, when you do the positive biasing protocol and then switch over, go to CBI2, will this activity be ingestive or not? And what Flora found is that this activity is ingestive. So the fact that there's been intervening egestive activity here has no impact. So this activity is as ingestive as if there was no intervening egestive activity. And you can see that in the sample recordings. This is radula closer activity, high frequency during retraction, without the egestive and then with the egestive, and you can see it in the data plotted below here too. Okay, if you're wondering why there's a difference between these two situations, why would there be a difference using these two different interneurons, B20 and B65? What it has to do with is that the fact that CBI2 makes different synaptic connections with these two cells. So CBI2 makes an excitatory synaptic connection with B20. So if something has happened, if the B20 excitability is up, and then you try to trigger a cycle of activity with CBI2, then CBI2 is going to produce some activity in B20. B20 is going to be overactive, and so activity is not ingestive. In contrast, CBI2 makes an inhibitory synaptic connection with B65. So if something has happened to increase the excitability of B65, and then you try to trigger activity with CBI2, it doesn't matter that this excitability is up because there's inhibition here, so B65 doesn't fire and the programs can be ingestive. Okay, so a conclusion then of Flora's work is that the circuit mechanism that's used to pattern activity can determine how readily task switching can occur. All right, so to finish then, in summary, We've shown that modulators that configure network activity during repetition priming have persistent effects. Our data strongly suggests that this has consequences for task switching. With task switching in one direction, persistent effects appear to impede subsequent task switching. It doesn't always work this way, however. So in the other direction, persistent effects appear to promote rather than impede subsequent switching. So in the one direction, the egestive to ingestive, you have this, it's impeded, right? You try to go to CBI2, activity still remains egestive. After CBI2, however, you can very effectively switch over to both egestive activity, that's a positive biasing, and you can even return to ingestive activity. So after the repeated stimulation of CBI2, you're actually in this situation where you can very effectively switch back and forth from ingestive to egestive types of activity without going back to the intermediate state. Okay, so my very last slide then, I just want to acknowledge the people in the Aplysia research group who contributed to this research that I didn't mention when I was giving the talk. And I also want to give a special thanks to my co-workers and collaborators of many years, John Jing, and last but not least, Claude Weiss, who's been a terrific mentor and a friend and a great co-worker for many years. So thank you. And this, by the way, is a crab trying to get into Aplysia.
Thank you, Betsy.
Sure.
That's a lobster.
Oh, maybe. You would know better than I.
It's
Honey
fine.
lobster.
Yeah. Cool.
Okay, so I'll stop sharing.
Hey, any more questions for Betsy?
I have a question for Betsy.
Hi, Dawn.
Hi, I was trying to find your picture in the group of everybody. There you are. I was wondering if the persistent actions, are they all at the excitability level, or is there a synaptic component?
Right, well, they're not actually all, no, they're not all at the excitability level. They're just, I was sort of simplifying things there, so that's kind of the major, but they're not all, no.
Okay, thanks.
All righty.
I have a quick
Thank
question.
you.
I have a question.
Go ahead, Omar.
Okay, so great talk. And something I found interesting was that the ingestive priming takes some time, and I guess there's a number of cycles that it takes until you get to complete priming. How does that relate to the time constant of full activation by the peptide? So has anybody looked at that, how long it takes for the peptide to fully exert its effect, and how that relates to how long repetition priming takes to fully occur?
No, nobody's looked at it in the detail that you're asking. We don't entirely know why it's slow. What actually happens with the release, if you stimulate, this is going back now to Paul Church's work. Paul Church did some work where he just kept stimulating with the same parameters for kind of a long period of time. And he found actually that the peptide levels continue to go up, even if you keep the parameters constant. So presumably the peptide concentration is continually increasing, which could be contributing. But we don't actually know, but that's correlative, right? We don't actually know whether it's so that would be a presynaptic effect, but we don't know. Also, what's presumably happening is postsynaptically, there's summation, and we don't really know actually. But probably there's a combination of things going on.
Okay, cool, thanks.
Yep.
Just a historical question. I was interested in the remark that he made that the people at Columbia were not interested in neuropeptide. Was that, I mean, where were we at this point? Or you, I don't think I was around then.
Maybe
Did people,
old.
did people just not, did people just not appreciate the malleability of circuits to modulation, or was it just specifically about peptides? Was there bias towards amines, or was there generally just sort of like no idea that sort of behavior is in large part done by switching circuit dynamics with things that work on a slightly longer time scale than synapses?
Okay, now remember at Columbia what a circuit is. It's one sensory neuron. So that's a part of the answer.
So it was that specific crowd.
All right, so it was a lot of work to even get more neurons.
By the way, none of these guys are here, so you don't have to be careful.
No, I don't care anymore. I don't care anymore. So, right, so there's no, I mean, Eve would give you some great answers to that question too, but there's, yeah, for them, there's no appreciation of circuits and, right, I mean, it's one sensory neuron, one motor neuron, and that's it. So yeah.
That's serotonin, Betsy.
Sorry, oh yeah, and serotonin, right. Oh, serotonin was everything, right, initially, right, right. So, yeah. I think it was just a combination, it was just it's just an attitude too. When you show that something is common, maybe, some people can take that as, oh, it's not very interesting. I mean, in some ways, actually, maybe something's common is more interesting because it's actually used.
Well, particularly if you didn't know about it before, so.
Yeah. Yeah.
Well, Betsy, thank you so much for that. It was a great walk down memory lane in a lot of respects. And, but it also, I forgot the amount of work that it took to get those measurements. I mean, it was really interesting to see it in perspective of today's work. But I have a question. So we're looking at peptidergic neurons now, but through the lens of gene expression.
Yeah.
And we're finding changes in gene expression going up and down, depending on context for peptides.
Right.
So do you have any sense of whether peptidergic neurons store up peptides or change their gene expression that doesn't correlate in some way with the peptide?
Yeah, I think your results are going to be very interesting to see. So we don't really, we have noticed, I mean, this is work that we're doing now. We have noticed that there's a difference. So you can get repetition priming in a number of animals. You can also not get it. And we don't know for sure what's the issue there, whether it's a peptide issue or not a peptide issue. But yeah, I wouldn't be surprised if we tried, we tried to do some very simple things, feed the animals. Is there a difference in the peptides? Don't feed them, and we didn't find anything, but we didn't look, it certainly could have been done better.
Well, with egg-laying hormone, we found that if you starve the animal, if you deprive, I'm not even saying starve, if you deprive them of food for 7 days, the number of neurons expressing ELH goes way down in Invergia.
Yeah.
So I don't know why. I don't know what effect that has on the behavior.
Yeah. So what we're doing now is, so there are other ways that you can make activity ingestive that don't rely on this. This is only one of the inputs. So we're looking at what happens when it doesn't happen this way and how other, this is just one solution, but yeah. Yeah, I think there certainly is regulation. And we always found there was a huge amount of variability. That's why everything had to be pooled.
So say that some more. I want to hear about that variability.
Oh, it's just variable, so the, the number, what we were quantifying counts, if I showed you plots, if I showed you our original data, like how much SCP was in B15 in one animal versus another one, it was very, very, very variable. And Fred also found when he was doing the RIAs and measuring peptides, the femtomoles of peptide per drop in one animal could be high, much higher than another one. So
So-
everything was plotted as percent release because we had to normalize the pool of the data.
But do you think it's animal to animal variability? Do you think it might be day to day variability? It's like if you were
Oh.
able to observe that animal over time, would it vary?
Oh, I don't know. We never did that.
Right. It wasn't
We
possible. It
ground
wasn't
up the
feasible.
cells. Yeah,
Right,
that was
right. You're
it.
done.
Yeah. Yeah. Yeah.
Okay, Smita and Nelly, do you want to announce the upcoming talks?
Yeah, next month is June, yes. And we are bringing Astrid Brains on Thursday, June 17th. So same time, 2:30 to 4 p.m. I would like to request everyone to pass on this Zoom information to your undergrads. I know they are not there yet, but bring more people, especially undergrads. And
Thank you. And I just wanted to tell everybody that I just got an email that Smita got a very nice fellowship. So
Oh,
yeah, thank you, everybody.
Yay.
Thank you, Smita, for all your work in organizing this.
thank you.
Great.
yes, thank you.
Bye-bye.
Thanks, Betsy. Bye.
Bye.
Bye, everybody. Thank you, Betsy.
Thanks, Betsy.